Greetings, friends, fellow Earthlings, and the results of billions of years of evolution on one small rock in the void. This is Ask an Astrobiologist, the show that celebrates the science and the scientists involved in our quest to understand the nature of life. I'm your host, Dr. Graham, the cosmobiologist Lau, and as always, we're brought to you by the NASA Astrobiology Program and SegaNet.org. Thank you so much for everyone for tuning in today. As always, we love to give a huge shout out to those of you out there engaging in conversation with me, with our guests, through the posts on the NASA Astrobio Twitter account, and for just sharing about our episodes and all the things that we're doing. This month, we want to give a special shout out to Dr. Francis Rivera Hernandez for sharing about the show. Dr. Hernandez was a guest on the show previously for episode 38, 20 episodes ago. She joined us along with Kenda Lynch and Svetlana Skolyar to talk about the Perseverance rover and the geology of Mars and what we're looking at and trying to understand if Mars ever had life or currently has life and what we might find there. But now with today's episode of Ask an Astrobiologist, we're going to embark on a journey into the past for life on Earth, as told in the tapestry of the signs that remain in the rock record. We'll be diving into the ancient Ediacaran period, a pivotal chapter in Earth's history that witnessed the awe-inspiring rise of animal phyla and a lot of experiments in evolution and biology, and extraordinary biodiversity was occurring, leading to what we currently see here on Earth now. Today's guest is a paleontologist and an expert on the Ediacaran period. The range that living things existed in during the Ediacaran was around 635 to 539 million years ago, at a time when animal life as we know it was rising onto this planet. Dr. Mary Droser is a distinguished professor in the Department of Earth and Planetary Sciences at the University of California, Riverside, and someone who has dedicated her career to unraveling the mysteries of the Ediacaran period. Her groundbreaking discoveries and meticulous studies of fossil assemblages have shed light on the early evolution of multicellular life. Dr. Droser's expertise has taken her to fascinating field sites around the world, including her own work in helping us to develop the South Australia's Niplena Ediacara National Park. Today, she joins us to share her invaluable insight and take us on a captivating journey through time as well as her own career. So, Dr. Mary Droser, welcome to Ask an Astrobiologist.
Great, thanks for having me.
Awesome. Thanks so much for joining us here. 1 second, I have to work my teleprompter here just for a minute. All right, thank you so much. One thing I love to ask our guests when they first join us is to share with us their science origin story, the thing that inspired them to pursue a career in the sciences in the realms of paleontology and other disciplines related to astrobiology. What was it that hooked you into a career in the sciences?
Oh, I was very young when I got interested in the sciences. When I was a little kid and like 5 years old, wanted to be a marine biologist because of just floating around in the shallow seas off Long Island. And then age 10, I sort of got interested in geology. And it's a boring history. The rest is history, combining marine biology with geology and paleontology. And I was a freshman geology major, which is not usual. Most people discover geology during college. So yeah, very early on, loved science.
I love that so much. I mean, for me, myself, when I first started college, I had no idea I'd be a geologist eventually. I wanted to do marine biology and some other things, and then just found geology along the way. I think that's the case for many of us. So what in your career then took you into this realm of paleontology and specifically studying the Ediacaran?
I got into sort of interested in how animals interact with sediments and interact with their environments. And that got me into sort of this world of looking at bioturbation and burrowing and mobility. And I just kept going deeper and deeper in time. My dissertation was looking at sort of the advent of mobility and when and how animals first started moving and changing the sediment. And it just kept getting deeper. And eventually in the Ediacaran, where mobility first originated. And so I've never left.
I love that. Well, I mean, life has certainly left since that period and moved on and done a bunch of other interesting things. And, life on our planet, we have good evidence, maybe 3.5 to 3.8 billion years ago. Some of us hypothesize maybe 4 billion years or even earlier, life was happening on Earth. But, much of our lifespan of the time of life on Earth was microbial life. We had a lot of evolutionary change and adaptation. We had the rise of oxygen thanks to life, but it really was during this Ediacaran period, that life kind of started really blossoming out into this more complex, multicellular, the organisms that we kind of know of today. What do you think really drove that complex behavior to drive this process forward to allow life to kind of emerge in that realm?
Right. Well, that is a big question, right? You need two things. You need the environment to be correct, and you need the genes. You need the genetic pathways for multicellularity. And there are a lot of people looking at the advent of multicellularity from biological and paleontological, many perspectives. So there's two ways in which we think this might have happened, and most of Earth's history has gone by before we get multicellular animals. And multicellularity arose in a lot of different unrelated clades many times. But in terms of animals, it is a question of, did we have the genes and we needed the right level of oxygen, and we didn't have that until the Ediacaran, is one of the sort of leading hypotheses that it all comes down to oxygen. We need oxygen. So that is what we think may have been that last push for multicellularity in animals anyway.
Yeah, absolutely. Excuse me for one moment. Sorry, I had a cord drop there. So we did ask our audience online. We gave a little poll about some creatures of the Ediacaran period, and asked them which creature in the images we shared wasn't from the Ediacaran. And it's an interesting image. So 3 of those are actual artists' renditions of the kinds of fossils that you've discovered looking into this period of time. One of them was an artist drawing of the facehugger from the Alien franchise of science fiction. And interestingly, we actually fooled about 20% of the respondents, did not know which was which. And so there were some really interesting things going on during the Ediacaran, where we had different, kinds of adaptations, different kinds of things that life was doing. How do you kind of envision what life was actually doing at that time, these new experiments that biology was up to?
Yeah, so my fantasy world is getting in Miss Frizzle's magic school bus and going back to the Ediacaran. When I think about the Ediacaran, or when I try to describe the Ediacaran, the first thing I do is talk about the seafloor. So the seafloor of the Ediacaran, there were no burrowers, going deep down. We had mobility, but nobody really churning the sediment. We didn't have the effective grazers that we have today, things like sea cucumbers and whatnot. And so the seafloor would have been covered with like ooze and the kind of thing you would never want to put your foot in, right? Think pond scum. So when you start to envision the Ediacaran marine environment, and we're in the ocean, of course, first you start with your seafloor and the sort of green and blue and brown and disgusting sort of microbial mats and organic mats on the seafloor. And then put on that organisms. So what we think, actually, you imagine people think, oh, were they rare and little tiny little things here and there? No, neither rare nor small, big and small. So you start with that seafloor and you put frond-like organisms that were as tall as I am. You put some things that looked like bath mats that could be, a meter long, that would roam along the seafloor. And, lots of groups of organisms that would have just sitting there like little buttons on the seafloor. And so if you were snorkeling around in the Ediacaran, you'd be like, all right, this looks like an alien world. It looks like something that somebody created, or some alien movie that takes place under the sea. That's kind of familiar, but isn't really familiar. And so what we're trying to do is do the deep dive, no pun intended, into figuring out what is there and how do they relate to life today. But it totally would have been an alien world, and they're not familiar. And when we show people the fossils, they're like, yeah, no, I have no idea what this is. So yeah, I'm not surprised that it was 20%. They are weird looking things.
Yeah, and I think if anything, it was more that people saw the facehugger and they knew that from Alien, rather than knowing that the others weren't actually artist renditions of some science fiction creature. Now, I have read of you that you've had the opportunity to name a few of these fossil creatures that you've discovered, including one that you named after your own mother, one that you named after David Attenborough. What's it feel like to have like these fossil creatures that we know of on Earth, that you had a chance to actually give them the name and kind of share that with the world?
It's crazy and exciting, right? So we've named, I think, 10 or 12 different organisms. What's amazing, right, is that these are whole new body plans. This isn't just a little tweak on something we've already known. These are new body plans. And the way in which and sort of why we've discovered so many is that we're not doing paleontology the way others do paleo. So I liken it like normal paleontology is like the clink clink method. You go out in the field, you're like clink 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 clink. You take the fossil back to the museum. Where we're working at what is now Niplena Ediacara National Park, we are able to excavate whole bedding planes that represent snapshots of the Ediacaran seafloor. So if you imagine, going out to the seafloor today and a storm coming and dumping a bunch of sand on the seafloor, and that sand becoming rock, and casting those organisms on the base, we're excavating these beds, turning them over, and literally have snapshots of the seafloor. So what we're doing is we're mapping these beds, and we're finding all sorts of new and different body plans that are unlike other things. So that's where it gets really weird. You're like, I have no idea what this is, and we give them nicknames that are sort of what they look like. So Attenboroides looked like a raisin. So when we first saw it, we'd be like, oh, here's a raisin, and in my logbook, it says raisin. And then we found a bed that was covered with them, and we're like, all right, we have to deal with this. And, by looking at it and doing all sorts of looking at the preservation and the nature of morphology and everything, we reconstruct it as, not to take the analogy too far, but as sort of a grape-like organism or a sort of ctenophore-like organism. And so that's what's fun. It's amazing. It's a privilege to be able to look at these new body plans. And so, yeah, it's totally crazy and exciting, and trying to figure out where they fit in terms of the tree of life. I have a lot of wrinkles from just that. So.
Yeah, I mean, there's a lot to uncover there, from those early ctenophores and sponges, and then kind of coming up to where we're at in the Ediacara. And you mentioned Niplena Ediacara National Park, which was created in 2021, and you've had a hand in the development of the park itself. Can you speak to the importance of that site in general, not just for understanding this fossil assemblage, but also that site in general and why it's such a compelling region?
Yeah, so we, I started working there about 20 years ago with my colleague Jim Gailing at the South Australia Museum. We knew about probably 2 or 3 years in that this was a special site. And so over the last 15 years, and very much so over the last 7 or 8 years, we've worked with the South Australian government, the pastoralists who own the property, the traditional landowners, the Adnyamathanha, to come together to recognize that this is a unique site, and it's a site where people can walk around, both to bring science to the community, which I think is hugely important, as well as to conserve and preserve the fossils. So it's a wonderful opportunity to have kids and adults and come and learn about fossils and learn that Earth has a history, and that going back in time can teach us about the future, is important to understanding our life, is important to understanding how we might look at life on another planet. And Niplena Ediacara National Park and the Ediacara fossils there are also part of a World Heritage bid that we're doing for the region, which is sort of the dawn of animal life, which will take it from early eukaryotes into the Cambrian, and sort of when we have skeletonization and predation and things like that. So we're looking at this, a couple hundreds of millions of years period where we can track the crazy environmental changes which form the backdrop of all of this, as well as these appearance of these evolutionary innovations. And so it's an astounding place. It is nearby is the GSSP, or global stratotype point, for the Ediacaran. Literally, there's a golden spike in there, and so it's an amazing site. And I think just bringing science to the public, and obviously for this particular community, it's also an economic help, which is not a bad thing. So.
Yeah. That's awesome. Well, I want to share a quote that we had from you in a NASA Astrobiology article that, simply put, this is the best record of the dawn of animal life on Earth, looking in this Niplena site. I'd like to connect this to astrobiology, though. How does this kind of impact our view of how life might change and evolve over time on other worlds?
Yeah, so I think, I'm sort of at the tail end of what's interesting, right? When we think about astrobiology, we think in terms of teeny tiny prokaryotes, microorganisms, and by the time you get to multicellularity, you're like, whoa, major innovations have already happened. But I think there are a couple things we're doing that I think that is important. One, looking at sort of rate and scale of evolutionary change and innovations. What we see at Niplena and the diversity and the biomass that comes along very quickly once conditions are right. So that's a really important insight into understanding how life might evolve. If the conditions are right, it's just going to go, and it's not going to, it's not rare. It's diverse. They do a lot of different things, and I think that's one really important aspect. What are the biological, physical, and chemical processes that get us to that point? And those are the kinds of things we're trying to get a handle on. We're also looking at aspects of sort of what does life do to the record? So the record of sediments that preserve these. So one of the things we're interested in is, I mentioned, the sort of widespread microbial mats. And when you have widespread microbial mats, that changes the nature of how sediments are packaged. And so we are currently looking at how we can recognize that from a cross-section and recognize, just without seeing a microbial mat or even the texture it is, but from the cross-section of looking at the side view of sedimentary rocks, how we might recognize that life was there. And we knew that there were certain structures that microbial mats imparted on sedimentary rocks, and that would be a mechanism for looking at life on other planets. But one of my graduate students, Rachel Sopranic, came in my office and said, I think we could do this through machine learning. That has led to us now developing, we've published a couple papers on this machine learning process of recognizing the types of sedimentary packaging that you get with microbial mats. Good lesson for always listening to your graduate students. They have great ideas. But also, there are multiple ways we can get at how life changes a planet and how life responds to planetary changes. And those are the kinds of questions we're going for.
Very cool. That makes me think a lot about like the Mars sample return process right now going on as well.
Exciting.
We bring these samples back, and if we have a side view of some of this sedimentary rock from Mars, that could be a huge step forward then using that machine learning process to look at those samples.
That's right. I look very keenly when pictures come back from Curiosity and Perseverance. So yeah, exactly.
Awesome. Well, so you mentioned the Ediacaran kind of as like this period, almost like an alien world occurring on our own Earth in the past. We did ask another poll of our audience on YouTube about the timing of the Ediacaran period. It started 635 million years ago, and we asked, how long was this period in general? And we had a lot of answers. The correct answer was 96 million years. Most people guessed 34 million years. There is some debate as to how the Ediacaran came to an end and how we get into the Cambrian radiation or Cambrian explosion then of life as we know it. What is the current thinking of how this period came to an end and we kind of lost some of those experiments?
Yeah, so the Ediacaran period was long, ranged from 635 to 539-ish. The Ediacara biota inhabit a part of that. So they are a part of the Ediacaran period. There's just so many things named after Ediacara itself. So I think, I have the perspective of somebody who works on the Ediacara biota. So in my mind, the Ediacara biota is the fuse. It's the beginning of the rise of animal life. And by the end of the Ediacara biota, we have most major body plans. There is a debate about what happened to elements of the Ediacara biota. My group would say that within the Ediacaran period, during the time that the Ediacara biota reigned, that there was an extinction likely due to a drop in oxygen. So that got rid of many of the things that are near and dear to my heart. But what happened right at the end of the Precambrian and the beginning of the Cambrian is the subject of debate, whether it was environmental changes or the beginning of all of the things that come around in the Cambrian, like bioturbators and predators, and did that cause the demise? And that the verdict is out on that. I mean, many people have very strong views, but there isn't a consensus yet. But a number of things in the Ediacaran do make it through. So it's not a failed experiment. There was a lot of experimentation, evolutionary experimentation, but some of these things did, in fact, make it through.
Very cool. And that's kind of a fun segue then, talking a bit more on the larger scale about life on Earth. You are a co-lead on one of the NASA astrobiology RCNs, or Research Coordination Networks, indeed the newest one called LIFE. I wonder, can you tell us the objectives of the LIFE RCN and what we intend to find through this RCN's work?
Yeah, sure. So LIFE is early cells to multicellularity. It's LIFE, all caps. And NASA's big on acronyms. This is not an acronym. So people always ask us what it means. And we're like, when we find out, we'll let you know, just because that's how we feel about LIFE. So LIFE is this newest of 5 research coordination networks, and it's focused on the coevolution of life and the environment on Earth. And so what we're looking at is we're bringing together scientists from all over disciplines. So, from molecular biology to experimental to paleontologists, biochemists, mission scientists, to look at how, to look at the nature of life's innovations and how that changed, what was the consequences of those, and how the chemical and physical processes on Earth impacted the nature and advent of these innovations. And I think, this RCN, which brings together, it's to foster community collaboration. But I think of it as sort of like your first day of kindergarten, right, or preschool. You're like, wait, what? There's all these other 5-year-olds. So we in these RCNs sort of bring together people who are studying, from my perspective, say, the advent of multicellularity from so many different perspectives. And these aren't people that I would normally interact with or even be on a Zoom with. And suddenly we're all brought together and we're in Zooms or in the same room discussing these major evolutionary innovations and getting out of our silos and talking, people who are studying, the advent of metabolisms with somebody who's looking at the oldest fossils. And it's so exciting. And, it would only happen in this sort of RCN framework. And it's, I think it's really bringing science forward much more rapidly than it would ever, even if we're like, yeah, I read those papers, but like, sitting down and talking to somebody is an amazing thing. And we're trying to bring together people who don't even know they're astrobiologists or doing astrobio. They may be doing modeling work or experiment work and not understand how relevant it is to what we're doing. So very exciting. And we have great early career scientists with us and steering committees. And so it's, we've been around for about a year. We have a new lecture series. We've had one, second one coming up. These are open. So we invite everybody to join us. And we certainly invite astrobiologists interested in life to join us as well. So very fun.
Awesome. Yeah, and that reminds me a lot of just astrobiology in general as a field. It really is not a single discipline. It's a large field of different disciplines coming together. I've often said one of the best and worst things we ever did to science was break it apart into all of these sub-disciplines, because you ended up with physicists in one building and chemists in another building on the same campus, not talking to each other. And now with astrobiology, with RCNs like LIFE, we have people communicating across those boundaries that were holding them apart before to share ideas. And so I really appreciate that viewpoint. So thanks for sharing that so much. Something I shared recently on my own Twitter account was an image about convergent evolution. And I asked my audience basically, what forms of life do they think, or what things do they think life would be converging upon for alien life? And there were a lot of answers. Some of my own favorite are things like trees. Amongst the crustaceans, we have several different convergences that get to the crab body style. Certain organisms like lizards losing their legs, or mammals losing their legs to become marine mammals. What are some of the convergent things in evolution that we see coming through the Ediacara and then towards the end that you think we would see with alien life as well?
Very interesting question. I think mobility, and not necessarily legs. I think long, squishy things that move, think worm. Mobility is such a game changer with respect to life. And because it allows organisms to seek out a new environment and or go get their food, right? And so I think, and convergence in a way to move your body whether it's legs, which we see, or convergence in dolphins and fish and marine reptiles all have a streamlined body so that they can move in the water. And I think those, for me, my immediate reaction would be convergence that allows you to move, would be my first off the cuff answer anyway.
I love that. Yeah, it makes me think a lot about what kinds of movement would be possible out there, and I like the idea of the little squidgy kind of moving structures in other alien worlds. Who knows if they ever found fins and legs and wings and those things? Maybe they just kept on moving, like worms and like little squishy things.
Yes, or
possible.
Big squishy things.
That's true. Yeah, giant squishy things. Who knows? That's some of the fun of thinking about what alien life could be based on what we know of life here.
Yeah.
I do want to move on to our audience questions very soon. So for those watching on YouTube right now, please ask your questions in the chat. We have moderators who are listening in to get those questions over to me. Just a few more things from me myself then. Before the show, we were discussing kind of, some of your hobbies and interests. And one thing you mentioned was that during your field excursions, you've taken your family along with you on your excursions, your children who've now grown up and are doing their own things. I wonder, can you speak to the importance of family life for a scientist as well, and the impact that family has had on your own career?
Yeah, no, great question. Yeah, I mean, I do field work, so I travel a lot. My husband works in the Himalayas, so, also away. So from when my kids were in diapers, they would come to South Australia with me, and I didn't really give it a thought at the time. In retrospect, I look back on it and say, wow, and my first 10 years, my mom came along. I would have nieces and nephews come along to watch my kids. And then over time, my two kids both got involved in the research, and we would get them out working in the field. And, nieces and nephews who came before just to watch my kids started coming and working in the field. And so kind of a magical for me to have had my kids grow up in this way. Literally, it was their summer vacations. I did wonder, would they hit 16 and say, I want to, go to the mall with my friends? And that never happened. One, my daughter is now studies ancient brines on Mars, is on the Curiosity mission. She's a grad student at Georgia Tech, where Francis is, of course. And my son is a first-year graduate student at Harvard studying marine microbiology and chemosymbionts and whatnot. So paleo adjacent, definitely not paleo, but and even nephews and nieces have gone into more science-related fields. So I think I am really, really, really lucky to have had that level of support from my extended family, as well as, to have been able to bring my kids in the field. And I do, it was very unusual when I was young and a new mother to do something like that, and people were horrified that I would do that and take my children to the outback. But wow, it came out well. And I do think that it's a way in way that we can visualize how we can parent and still be scientists at the same time. That's kind of an extreme, but there are other examples of how we can do that. So
yeah, I love that so much. I think during the pandemic, a lot of us, we had our children with us during our Zoom calls and our meetings, and I think it became kind of more acceptance in some ways for some people.
Yeah.
That, we also have our own families and our own lives outside of the science that we're doing. And
that's right.
If we can include our families in our science and our lives, that's all the better, because it gives us a chance to kind of enjoy the community of having everyone together and learning and sharing in that way. So I'm so thankful for you sharing that with us. We are going to move on now to our Faster Than Light segment. This is where we have a few simple questions requiring simple answers, hopefully, about some of the things that we wonder about astrobiology and the history of life and things like this. The first question comes from the idea of Fermi's paradox, which is more of a question that, where are they? And so my question for you is, what is your favorite answer to Fermi's question? Where are they?
Far away.
Good answer.
Space is big.
Yeah, absolutely. That was the big thing. I think a lot of people say it's just so vast. Can they even travel that fast across space and time? How much time would it take to travel tens of thousands or hundreds of thousands, or from other galaxies, millions or billions of light years?
Yes.
The truth is we don't know.
Millions of years.
Yeah, a long time. So here's one that's near and dear to my heart, since I love science fiction and fantasy so much. I love reading about what's possible and thinking about human stories. What kinds of stories have inspired you to want to explore more about life?
So I'm going to go back to my childhood again. So I was, and this is TV show. So story. So as a little kid watching Star Trek, and I remember an episode, and they're on a planet, and there are these rocks that are like, it's a rock organism, and Captain Kirk is really like, what, and Bones goes, I think it was Bones, might have been Spock, is like, that's not a carbon-based organism. That's a silica-based organism. And I remember being like, wait, there are silica-based organisms? And like, I was like 5 years old, but honestly, that has stuck with me as sort of this way to conceptualize life on other planets, and sort of watching this science fiction show and just being like, wow, that changes my view, So anyway, a little bit, not some great read, but rather just having watched a TV show as a very little kid.
Awesome. I think Star Trek has impacted a lot of us who are in the sciences now.
I still
so that's
love
awesome.
it. Still love it.
So you've had a very prestigious career. You've done so many incredible things. But if you could take the knowledge you have now and go back and meet yourself at the beginning of your career, what kind of advice would you give yourself?
Yeah, as you say, I've been very lucky. If I were, I would say find your people. And I was really lucky. Past me was very lucky in terms of finding my people. But I think what I'd give advice to younger faculty or early career scientists is, find your colleagues, your students, junior and senior to you colleagues, your family, find your support group, and find your colleagues that you respect and trust, and who trust and respect you, and hold on to those people. And I think it is about the people you interact with, not the institutions you go to, but really, who is it that you work with that you're learning from is awesome. Really important. And I was very, very lucky on that score.
I love that. What is something that excites you about the future?
Gosh, right now, I would say the early career scientists getting into astrobiology. So when I was young, astrobiology wasn't a field. I mean, I was like mid-career before astrobiology was a field, and you have your graduates. Right now, there is AbGradCon in San Diego, which is bringing together graduate students in astrobiology. So it's the graduate students, junior faculty, young mission scientists, and, coming together and bringing so many different perspectives and diversity of backgrounds and scientific backgrounds and unconventional thinking. And, it's just bringing new voices to the table, I think, is so exciting. And, just coming at it from a very different perspective, and different than, starting off unsiloed. So it's not saying, oh, we're bringing in molecular biologists. These are starting off unsiloed, which is terrific.
I love that. Good shout out for AbGradCon. I was a past member, eventually co-organizer, eventually lead organizer for AbGradCon.
There you go.
And I love that, everyone's learning all of these different disciplines together. There aren't different sessions. Yeah. It's all one session where everyone's joining in together.
It's fabulous. AbGradCon, huge fan.
Absolutely. So what is the best part of your job?
That I get to work on the Ediacara biota where it was discovered. What a privilege. And obviously the people I work with, students and colleagues and family that I get to work there with. But I go back in 3 weeks from now, and I get like antsy. I can't wait. I get out in the field and I'm like, how is it that I am so lucky to sit here and uncover these fossils? And honestly, and this is my job? That's crazy.
I love that so much. So one more question for our Faster Than Light segment. It's my favorite question of all. What is one scientific fact that still blows your mind?
From a biological perspective, I would say it's the commonality of developmental controls among animals. And this is in my lifetime that we've figured this out. The fact that there are similar genetic pathways and genes involved in the body organization of all animals, and disparate animal plans. So we can, that idea of the same pathway, we wouldn't have ever imagined it. And it's a way also of thinking about and interpreting the Ediacara biota. I think, wow, that's cool. So yes, I mean, the number of exoplanets also very, very cool. But the sort of genetic pathway and common theme, that's amazing.
Absolutely. I love that so much. We are going to transition now to our audience questions. So for those watching live on YouTube right now, you can ask your questions in the chat. We'll try to get as many as we can in the teleprompter. I see we have a large number already, but we wanted to start with kind of a fun one. So as a geologist, I think any of us who've done any kind of geology, at some point, someone's going to walk up to you and say, here's a rock I found. What is it? I've had this happen with family members with what they think are meteorites. I've had people I don't even really know bring up rocks to me and say, what is this? And we had a user on Twitter reach out for our questions, and they wanted to know if you could identify a fossil for them. User Brindley Ash on Twitter said they found this fossil in North Alabama, and I think we have a picture of it going up right now. And their question for you is, could you identify this fossil from this image?
So thank you, Brindley. I love this kind of thing. So I did take a peek. And so my first thing to say is, I'm not sure, right? This isn't like it's a trilobite. What it most reminds me of and what my first gut would be is that it's a conularid, which is an extinct organism that had four sides, sort of like a four-sided pyramid, but a cone. If I'm looking at it correctly, it looks like it's sort of on moss or something, which gives me a scale of about that big, which would be consistent with conularid. It looks like it has some pustule things around the edge. Not sure what they are. Conularids, we don't think had pustule things around the outside, so I could be completely off and it's some part of a vertebrate. But that would be my first port of call. What I would do in this, and if it's a conularid, they have phosphate plates, and this doesn't appear to. So then we take it the next step and say, is it a mold or a cast? Like you would have a cast on your arm, and so this would be the outside of a conularid. We'd look to see what age rock it's in. Do we have other specimens from there, and so on. But thank you for this. That would be my first gut on that.
Love it. That's great. Yeah, because it's hard to know unless you have it in hand, and even then you have to do some work sometimes to figure out exactly what you're holding in your hand sample as well.
Yeah, go find some more is what I'd say.
There you go. Our next question comes from Jim Pass on Twitter. Jim has, first off, a question that I think I can handle here a little bit, asking about risks to humans over time for exploring Mars, assuming that there might be viruses or bacteria there, and whether or not they can hurt us. And I think the immediate answer is, well, we really don't know. If they have a different origin and lineage than us, then they're probably less likely to actually be able to hurt us or interact with us. If they have the same lineage, though, that actually could mean that they have some potential to do some harm. But there's such a wide range of possibility that we really don't know. However, Jim has a second part to his question that I think is actually kind of interesting here. He wants to know what the status and roles for an astrosociologist could be in doing these kinds of missions. And given with the Niplena Ediacara National Park, it's not just about the fossil bed. It's also about the people and the culture and the history. I wonder, do you have a vision for how sociologists fit into the work that we're doing in astrobiology?
Oh, I think in many different ways, I would say. In terms of Niplena, it is the traditional lands of the Adnyamathanha people, and that is very, very important to us, and will always be the traditional lands of the Adnyamathanha people. And I think that is something that is important to us and important for us to respect, whether on Earth or, in fact, on Mars. In terms of working with people, you can't work in science without working with people and understanding the culture either of the area where you're doing field work, or in fact, asking the difficult questions about, whether you're doing experiments or thinking about going to another planet. And of course, we do worry about that in terms of the impact of going to another planet, also on our own culture. So yes, absolutely, unequivocally. Astrobiology isn't just about science. It brings in all sorts of disciplines, including sociology. So yes, absolutely.
Awesome. We have two questions that are fairly similar. One from Accelerator712 on Twitter, the other from Hilbert Spectrum on Twitter. The first one was what was Earth's ambient temperature during the early formation of animal life? But connected to that is this other question of can paleontology tell us about the history of the environment and the environmental history during periods like the Ediacaran when these things were changing so much?
Great question. So the Earth has had vastly different climates than we do now. When the dinosaurs were around, there were no glaciers at the poles, for example. It's been a lot colder and a lot hotter. And some of that evidence comes from the fossil record, and a lot of that evidence comes from the geochemical record that's preserved in rocks. So in terms of the ambient temperature when the Ediacara biota were around, the base of the Ediacaran period is marked at the base of at the end of what we call snowball Earth, which is the time we've evidenced that there were glaciers actually down at the equator. And, whether the Earth was a snowball and completely covered in snow and ice, or whether it was more of a slush ball and oceans were open, we don't know. But that's a really different world than we imagine today. In terms of by the time the Ediacara biota first appear, they lived in very different environments and at different latitudes. So the oldest Ediacara biota lived in the deep sea, not the ones at Niplena, but older than that. That would have been cold, no matter what, colder than the shallow parts. They spanned certainly from the equator to well into the higher latitudes. So the Ediacara biota would have experienced a lot of different temperatures, but we were past snowball Earth, and in fact, they first appear after a second sort of glaciation. So I don't think they would have. They weren't around during times of glaciation, but the fossil record is one of the greatest insights into understanding past climate change, which helps us, of course, understand and constrain future climate change, and understanding the past and how organisms responded to past climate change informs future climate change. So great questions.
Awesome. Larry Ryan on Twitter has asked what your most revealing or maybe favorite discovery has been so far.
Oof, so many. I have to go back to sort of scientists rarely have light bulb moments. Even when I uncover a new body plan, we're like, yeah, is that really a new body plan, or what is that? So we've had the excitement of discovering new body plans and fossils, some of which we're still discovering. One of my most favorite moments was actually I had two different fossils that I was trying to figure out, and I just didn't understand them. And literally having a light bulb moment with a particular slab of rocks when I could put them together and realized one was the holdfast of the other. So, the sort of root of the other. And this ultimately became Phoenicia. And we found lots and lots of similar-sized holdfasts together with these similar-sized Phoenicias. So that told us a lot about their reproduction. I'm going to make this long story very short, but basically evidence of sexual reproduction. And so it was our oldest sort of pretty definitive evidence of sexual reproduction in animals. That was really exciting and led to some very fun and sort of crazy media after that. And you can imagine sort of the roads in which different organizations picked that up. But that was really a fun, literal light bulb moment of, oh my gosh, these two parts go to the same animal. That was great.
That's awesome. Yeah, and like the evolution of sexual reproduction, this ability for organisms to swap genes together like that allows for so much more biodiversity.
Yes,
and so much more potential for evolution. So that's a very huge moment. I love that so much. We have user Jared Brunk on Twitter wants to know first about Europa Clipper and possibly finding life, and then also what you think about Europans in the Europa Ocean, possibly being anything similar to what we've seen here for the animal life on Earth. I will personally start off by saying that Europa Clipper is a wonderful mission. It's not going to be looking into the ocean of Europa directly. It'll be monitoring the surface by orbiting around Jupiter and Europa together, giving us an understanding of the chemistry of the surface, helping us to understand the depth of the ocean. It might have a chance of sampling some fluid if there are plumes coming out of Europa. We don't know if there's there yet or not for sure. But it's not going to immediately be testing the ocean. However, Dr. Droser, what do you think of this possibility for there to be a European biosphere, anything like Earth's?
Yeah, I'm very excited about the Europa mission because, of course, we think icy worlds, ocean worlds. We're an ocean world. We have ocean life, as far as we know, originated in the ocean. So I'm very excited. And, I think what we're trying to do there, and the same thing that we do when we go back in time, is constrain the environment. Was this an environment that might have been suitable for life? And I think we'll get a lot of those answers from this mission, not necessarily, hey, there's a Dickinsonia on the seafloor. In terms of what might be there were we to find life, I think we're If people ask me about, oh, are you going to find Ediacara fossils on other planets? And my answer to that is, no, I don't think we will, because evolution would, by chance or different conditions, take a different pathway. So we might find life there. We might find microbial life or very, very simple life, but it's understanding the chemical and biological processes that might give us that life that I think is really interesting and that we're going for. I'd love to find an Ediacaran fossil on the seafloor of Europa. Pretty sure that's not going to happen, but it would kind of be a big day for me.
Yeah, I think it'd be a big day for all of us.
For all of us. Oh, yeah, it would be a big day. Any life would be a big day.
Absolutely. So coming back to Earth now, we kind of mentioned this idea of understanding some of the Earth's environmental record, temperature record through paleontology. We have two fairly similar questions. We have one from Rendering Reality 3D Animations on YouTube, asking about the chemistry of the seawater during the Ediacaran. But then we also have user SL on YouTube has said, they're a UC Berkeley biology major, planetary science minor, and they want to know if the Ediacaran correlated with the Cambrian explosion, Cambrian radiation, and the GOE, which we kind of discussed already. Can you kind of give us a vision then of kind of the understanding of Earth's changes leading through to the Ediacaran period?
Yeah, okay, so yes. So the GOE was about 2 billion years ago, and that's the Great Oxygenation Event. And it's the first time where we really sort of reach high levels of oxygen and or higher levels of oxygen. And between then and the sort of when we see the Ediacara biota, oxygen changed a lot and shifted. And there are many people who are trying to really pull apart and tease apart the history of oxygen during that time and how it relates to, say, the first appearance of eukaryotes, which is a very big event, obviously. In terms of sort of the seawater during the Ediacaran, as we come up to it, the big part of the chemistry of the seawater we're interested in is the oxygen. And as I mentioned earlier, organisms need oxygen. The Ediacara biota would have needed oxygen. There's a lot of evidence that oxygen increased right before the advent of the Ediacara biota, and I think we're not surprised about that, and we think that that was a factor, and that even the history of the Ediacara biota was tied to changing oxygen levels in the Ediacaran. And we tease apart the oxygen from geochemical proxies that give us an idea of this. But the Cambrian radiation happened after the Ediacaran period and after the Ediacara biota, many of which would have been extinct before the sort of Cambrian radiation. From my perspective, I think of the Ediacaran as just the beginning, the sort of fuse that led to the Cambrian radiation, and that there were extinctions within that, but it was one continuous sort of radiation. My husband works on the Cambrian. He likes to think the action was in the Cambrian, but I think of it as really much more of a continuous radiation of animals that was with a backdrop of very dramatic changing conditions on Earth, many of which had to do with oxygen, some of which had to do with physical changes like sea level change, the advent, changing biochemistry, advent of skeletonizers and what that does to ocean geochemistry. So lots of changes going on during this time. Great questions.
Awesome. User Blueless on YouTube. First off, they're a geology student and said that they love the idea of exploring life and rock interactions. So yay, geology. How important are microfossils for astrobiology?
Oh, huge, huge. I mean, microfossils are how we understand life before we get before we get like the Ediacara biota and multicellular algae that we can see. So, to be honest, for most of our record, it is the record of microfossils, whether they're microfossils of eukaryotes or microfossils of prokaryotes. So hugely important aspect of our understanding of life as it evolved on our planet. We need to understand our planet first. I mean, we have a record, we can understand it, and microfossils play a very, very important role in that.
Absolutely. We have another two very similar questions. I'm going to bring them together. First off, Sean M. on YouTube wanted to know if there was any indication you've seen so far for the Ediacaran that there were the right conditions for evolution were localized kind of to the regions we've seen so far, or if we think what was happening during the Ediacaran really was planetary scale. And then connected to that is Arunava Padar, who has been a past researcher with Blue Marble Space Institute of Science, is currently a graduate student in Svalbard. So he's made a big move from India to the high Arctic, and he wants to know if Ediacaran fossils have been found in the Arctic. And if so, are they different from places in lower latitudes? And so I think those two questions kind of merge well together. What was the global extent of the Ediacaran to our knowledge?
So as far as we know, they were expanded globally. These are soft-bodied organisms. So right off the bat, you're trying to preserve soft-bodied organisms in the fossil record. That's hard. There were a number of conditions in the Ediacaran that led to ways in which it was easier to preserve soft-bodied fossils then than it is now. But the same fossils that we found, many of the same fossils that we find at Niplena are found, say, in the White Sea of Russia. And even when you reconstruct paleotectonics, that they were not close together. So we think they were pretty global, and that it is a function of the rock record and the right environments of the time, and then preserving those rocks now over 500 million years later. We do have a high level of endemicity, so there were some things that were very local, but we have other taxa even at the species level that were all over the place. In terms of Ediacaran fossils at the Arctic and the Arctic at the time, we don't have evidence of that because we don't have fossils from places, we don't have rocks from places that were actually at the poles during the Ediacaran. So again, that comes back to the rock record. We have a handful of really good localities of Ediacaran fossils. But we have scraps, then that's not a very nice word to say about fossils, but bits and pieces of fossils at a lot of localities around the world. And that helps us understand their distribution. Even if we find, a single part of one that we can identify, that still puts an Ediacaran fossil there. So Wonderful. Also great questions.
Yeah, and I think for Arunava, it could help to go online too and just look at how the continents themselves have moved over time and where continental things were at that period of time as well. It might help you envision then kind of what the world looked like at that period of time.
Absolutely.
We have a question from user AC on YouTube. They want to know, and I guess this is kind of going into genetics now, if we can connect the genetics to the fossils. They want to know how body plans of organisms evolved along with the genetics as controlled by things like Hox genes. In your work and your collaborations, do we have good ways to tie our knowledge of the genotypes of organisms and changes in genetics to the microfossils that we found?
Yes. What I'd say is, one of the things that we can do with Ediacara biota, and one of the ways that we're starting to think about it is, what can we say about these organisms? Were they bilaterally symmetrical? Could they move? And what does it take to do that? How did they eat, for example? And so what are the genes that are required in mobility? What are the genes that are required in having a nervous system that tells you to move, go after, there's good mat over there, and to go after that or go after a dead animal. So in that sense, we're starting to do that sort of evo-devo work with certain types with certain taxa that we have in order to sort of, we understand them better, looking at them from this perspective, as well as understanding where they fit in the tree of life. Can we constrain it? Was it a true animal? Was it a eumetazoan? Was it, do we have any idea? And so I think we're never going to know, but I think by using what we understand about modern evo-devo, we can then start to think about these questions for the Ediacara biota, which I think is a really important way forward in understanding how they fit and how these crazy body plans that don't look familiar fit on the tree of life.
Wonderful. User AC similarly had a question of what determines body plan. And that is an interesting question that connects to exactly what you just said, kind of connecting to, evolution development and connecting to our genes. But then we have this change in body plan. And so I want to expand that a little bit and kind of, again, speculate about possible alien life out there. Do you think we'll have similar body plans to what happened here? Or does some of the things you found so far from the Ediacaran suggest that there could have been much different body plans leading to larger scale organisms like us?
What I'd say about the Ediacaran is we look at different body plans, but in a way, they're all variation on a theme. So going back to mobility, if you move, you have to have certain constraints. We're bilaterally symmetrical, which helps us move and so on. We have body plans in the Ediacaran that we don't have today that are, we have triradial symmetry, right? Which is, there's nothing today that's triradial, and people will, I'll get a bunch of replies and they'll say, oh, but I found this and it's triradial, but really genuine triradial body plan. We have a pile of things that have triradial symmetry. That is an extinct body plan. But it is an organism that sat on the seafloor and probably suspension fed and whatnot. So I think I think it's sort of both ways. There are certain constraints that we have in terms of animals doing certain things. On the other hand, we have pretty significant body plans that are no longer around today. So maybe we'll go to another planet and find something with very, very different symmetry and so on. So I'm very open-minded.
Yeah, I think you have to be when you see all these weird things that life was doing and what could be possible.
Exactly. Yep.
It's so fun to think about. I will ask one more question from the audience. This comes from one of our production assistants, Anurup Mahanti. Anurup wants to know why paleontologists and geologists like ourselves define time looking back in millions or billions of years ago, I guess, as opposed to years from the beginning. And won't that scale make things tricky for geologists millions of years from now?
Woo, great question. Yeah, I mean, I think it's how we communicate, right? So now is what we know. And of course, as we have gotten better and better over the history of geology, right? Going back 200 years when we didn't know how old the Earth was, the logical place is to start now and look back. And it's both a way of communicating as well as just showing our increasing understanding of geologic time and how old the Earth is and how we have learned that. I look forward to geologists millions of years having trouble with this issue. I think that would be a great problem to have. So, yeah.
Yeah, I'm hoping that there are geologists millions of years from now.
Exactly. That's what I'm thinking.
Yeah. So, for our audience who are watching, with Dr. Mary Droser here, we talked about the Ediacaran, how, this was really an alien environment, and it gives us an idea of what other alien environments might be out there right now. One thing I've been discussing a lot with people lately right now is the emergence of these large language models and how machine learning and artificial intelligences are really taking off right now. And it makes some of us wonder whether the first form of alien intelligence with which we communicate might be one of our own creation. Do you think maybe these large language models and AIs that we're creating right now will become the very first aliens that we meet? If you have any input on that, feel free to reach out to me. I'm at Cosmobiologist across the interwebs. You can reach out on the @NASA Astrobio Twitter account or go to seganet.org and drop us a line there. We'd love to hear from you and hear your opinions. I also want to say, if you are interested in learning more about the Niplena Ediacara National Park, there is a website from the National Parks and Wildlife Services of South Australia. It's a really beautiful website. There's some information there you can find, maybe even if you want to go and tour the area yourself and see this national park in the future. Of course, for those who are tuning in right now live or watching the YouTube video later, if you want to sign up for the NASA Astrobiology mailing list, you'll get information about our show, about articles being published by NASA Astrobiology, many other events, conferences, meetings, ways to get involved in this realm of astrobiology being conducted through the NASA Astrobiology program. So Dr. Mary Droser, thank you so much for joining us, for all of your insight, and for answering the questions our audience had for you.
Thanks for having me.
That's been great. And we do want to give a special acknowledgement as well to credit for some of the images and video we've showed to Sam Oster, Robert Lang, and the South Australian Government Department for Environment and Water for sharing those images with us so we could share them with you as well. So to our audience tuning in, as always, thank you for joining. And until next time, stay curious.